Who can tell me what a coronal cut looks like? What's the view that we're looking at?
Right through the lateral thing.
Yes, so it's like you're looking straight into the brain, like face on. So we're doing cuts that are perpendicular to the floor, right? So we're slicing this way through the brain, from the top to the bottom, right? You have some coronal pictures in your human neuroanatomy book, and I'll go over those at the end after we go through the sheep brain stuff. And then we have two that are very similar pictures, slightly different, but pretty similar. Horizontal cuts. So a horizontal cut to the human brain would be a cut like this, right? So that you're looking straight down, so that you have anterior, posterior, and left and right, and you're looking down in the brain. So all of these structures that we're talking about today are what we call subcortical structures. They're not on the cortex, they're not on the midline, they're deep inside the brain, and so surrounded by tissue. So the only way to see them is through horizontal coronal cuts. So we'll have these two, and I'll go over those as well. I'm going to do a couple more. I'll probably do two cuts here, so I'll kind of run through everything twice. And then I made coronal cuts for everybody, so every station has a coronal cut brain in a Tupperware jar there, and it's in order obviously from front to back. What's real important today, like I've been saying in the past, is that you look at lots of different brain slices. So you may start out with yours in your station because you know the order that it's in, so you can kind of tell front to back. But when you go in for the practical, you're just going to have a slice, right, that looks like that, right? And you're not going to be able to see from other pieces if it's the front or the back. So I'm going to teach you how to orientate and how to know if we're in the anterior, posterior part of the brain and what the landmarks are. Okay? Good. So it's important. You can rehearse with the whole brain, but it's important at some point today to practice just random brain slices. And Madeline, which bucket over there has just coronal slices in it? That one? Does it say? No, it should say.
Top. Yeah, it says coronal top.
Yeah. So that small bucket there, it has just probably 100 just random pieces of brain that are all mixed up. So you just pull out a slice. Can I say is this anterior, posterior? How do I know that sort of thing? All right? All right. Good deal. So we're going to start with the whole brain here. Is this frozen? Oh, there we go. All right, we good? All right, and so I'm going to make my first cut. It's actually going to be, so here's the cruciate fissure across there, cruciate sulcus. So my first cut is going to go straight down a little bit behind there. Okay? And we'll look at each of these as we go. Okay? Now, we're in the anterior part of the brain, right? So I just cut right behind the frontal lobe. So this is what we would consider anterior. Is that better? All right. How do we know? If you just see this view, how would you tell if it's anterior or posterior? The way that we're going to know that is by looking at the ventricles and the corpus callosum and the fornix. Those are going to be your three key landmarks, okay, for orientation. Ventricles, corpus callosum, and fornix. Okay? All right. So here in the anterior part, we have the lateral ventricles, and we would call these the anterior horn of the lateral ventricles. So we're differentiating between anterior and posterior of the lateral ventricles. The lateral ventricles are the only ventricles that you can see, right? What's this thing in between them?
Anterior, maybe? No.
Septum pellucidum. That's the thin membrane that we saw in the midline cut that separates the lateral ventricles. Okay? This upside down U structure at the bottom of the septum pellucidum, those are the anterior columns of the fornix. So remember, the fornix on a midline cut all right, this is the corpus callosum, right? And we have here the rostrum, genu, body, and splenum of the corpus callosum. Remember, the fornix sort of came up like this, right? Sort of came up like that, like arching under. And then this was all septum pellucidum there, right? So what we're looking at here is a slice through the fornix here that creates that U shape, right, where this is the midline, right, right through that structure there. Does that make sense? Okay. Now, underneath here are actually the mammillary bodies. If so we were to flip the brain over and look at the ventral side, you have those two mammillary bodies that are right next to each other. So the way this all connects and works is that, can you tilt the camera so you can see me? Just spin it like that. There, good. All right, so if you can imagine that my fists are the mammillary bodies, right? My arms are the columns of the fornix that come up and make that U. The way this works is that my fists are down here, the columns come up, make the U, and then my back is where the fornix goes up against the corpus callosum and is high and flat up next to the corpus callosum. We'll see that in the posterior cut. And then the fornix wraps around and becomes the hippocampus. It makes like a U shape and comes back around. So my legs, so if I'm like this, my legs then are the hippocampus coming back around out the bottom of the brain. And it makes like almost a complete circle around the brain. And this is all involved with the motion and the limbic system. All right, so we're good on that? Before we get started back, let me just pause for a second. And I drew a little picture over here, so let me kind of go over that. This is what we're going to see in the anterior part of the brain. Which I think may be a little bit more complicated, or at least more structures than the posterior part of the brain. So, fornix, low and upside down U, okay? Septum pellucidum, anterior lateral ventricles, okay? Corpus callosum up here at the top. Yes, Becca.
Can you go to the second piece?
Yes, this line separating the anterior lateral ventricles, because it's like a sheet through there. It's very thin, okay? Then over here, right next to the anterior lateral ventricles, is the caudate. There's like a ribbon of white matter or axons, connections. It's a ribbon of connections there called the internal capsule. And then the outside has another gray matter that's also kind of like a hemicircle or lens-like shape. It's called the lentiform nuclei. And that actually has the globus pallidus and the putamen in it, but we can't see those distinctions in the rat brain. So in the sheep brain. So we just call it lentiform nucleus. It's a cluster of internal and external globus pallidus and putamen. These areas are all part of the basal ganglia, and the basal ganglia is involved with regulating movement. So this is the area of the brain that gets affected in Parkinson's disease, where we have the shuffling movement and the lack of emotional expression and cognitive deficits. It also is involved with regulating our thought patterns in our head. So it can be involved in obsessive compulsive disorder where people can't turn off obsessive thoughts without performing a behavior, a compulsive behavior. And so, so that's the importance of knowing where these structures are and what they do. Again, anterior part of the brain, we know we're anterior because the fornix upside down U, septum pellucidum, and the two anterior lateral ventricles. Okay.
So which parts of the visual ganglia are white matter and gray matter? Is it all three?
The internal capsule is white matter.
Okay. Okay. Or is that what you were pointing out right before the mouth on the brain?
Yeah. And so we will look at that again. We'll try and get some contrast there. This isn't a great example, but you can kind of see white matter there, and then this would be caudate, that would be lentiform, there's your fornix. So let's take another slice, and if we need to cut off the light so we can try that, but we'll take like a thin slice through here. This is kind of actually moving further back. So now the fornix is not really a U, it's kind of a big structure there. So this is not a slice that I would actually use. I'm either going to use a slice that's clearly anterior or clearly posterior, but not in the middle. Because I'm not trying to trick you guys, and it gets harder to see this. So I probably wouldn't use that slice. Let's keep looking and see what we see on back here. And I'll do another brain. We'll look at a couple different slices. I'm just going to make a couple more cuts here so you can see how far back. Okay. So we'll come back up here. Now, remember I said that as we move further back in the brain, I'm not sure that's better. I think we need some light in here. Okay, we'll work with it. As we move further back in the brain, the fornix now is coming up, and it ends up high and tight next to the corpus callosum. And so here, now, in this slice, we see the fornix is appearing as like a flat structure, almost looks like it has wings. Can you see that? Here's the corpus callosum. It stays a very distinct white fiber band. And you can see how the corpus callosum then comes out and connects all these areas of the cortex and white matter out here. Okay? So here's the fornix, here, corpus callosum. Okay? So we know we're in the posterior part of the brain. This is now up here in between the fornix and the corpus callosum, the posterior horn of the lateral ventricles. So the lateral ventricles are now high and in between the fornix, which is kind of flat, it looks like it has wings, up next to the corpus callosum. Down here below, this T shape here is the third ventricle, a midline structure. And on either side of the third ventricle is what? The thalamus. Remember last time on the midline cut, I had to stick a pin in either the thalamus or if I called it CSF, it was third ventricle. So now instead of having caudate or lentiform nucleus over here, this is thalamus in the posterior part of the brain. If we go further back, again, you can see the T shape of the third ventricle. This is thalamus, fornix, corpus callosum, What is this? If I had a pen stuck here and it said gyrus, what would that be? The cingulate gyrus. It's the gyrus on the midline just above the corpus callosum. Okay? So that would be right here. But now we're looking at it from a minute. And if it was stuck here and said sulcus, what would it be? Cingulate sulcus. Okay? You getting this? You sort of forming a 3D model in your head how this works? This stuff over here that's gray and looks funny in the posterior lateral ventricle, that's the choroplexus. The choroplexus, which makes CSF. If I flip this over, we go even further back. Now we're starting to have the fornix wrap around and become the hippocampus. So this is the hippocampus down here on the bottom of the brain. And it wraps around the thalamus to the hippocampus in the posterior part of the brain.
So where is it at the campus?
Right down here. Because remember, if we put this all back together and we flip it over, right, this is the hippocampal gyrus right here.
So signs that it's a posterior cut would be a hippocampus and the third ventricle.
Not so much the hippocampus as a sign, but the third ventricle and the fornix is high and flat against the corpus callosum. So the cue is the fornix. Is it an inverted U and you see the septum bluesum and two lateral ventricles on either side? Or do we have the fornix high and flat and a third ventricle teeth? Then we know we're posterior. Those are your cues. Again, this is going further back, and you can kind of see how this wraps around. The fornix is dying out and wrapping around. Now, oh, you can really see it here. So this is what used to be the fornix. See, here's the corpus callosum. You guys see that?
Is it white?
Yes, the corpus callosum is white. The fornix here is white, but it's turned into now this gyrus that wraps around that's become the hippocampus over here. See how it wraps around? What is this singular structure in the middle of the brain? We're only talking about one structure that there's one of in the middle of the brain, the pineal gland. Right? The pineal body or pineal gland. It's a singular structure on the midline. What about this hole here that's no longer a T but now is like a pipe? It's no longer a T though, it's now like a pipe. Where does the third ventricle go? The cerebral aqueduct. Right? So when you can see the pineal gland, you really know you're posterior in the brain, right? Fornix is turning to the hippocampus, and we have a pipe for a ventricle, a hole, which looks like a pipe instead of a T. That's cerebral aqueduct. Okay? And I can mark that today. Again, looking at the very back part, see the wraparound, pineal gland, and we have that hole right there. It's a really tiny hole. That hole right there. If it was stuck in there and said CSF, I'll still say CSF if I'm marking a ventricle, right? Okay. Good?
So if you label, can you label the hippocampal gyrus from that view? Okay.
Ken? Okay. Let's look at another real quick. I know we lost some time there, so we're going to pick up the pace. So you don't need me telling you this over and over again. Now, you can see why it's really important that you look at a lot of brains, because every time I make a cut through here, it's going through a different part of the brain. And the angles are different, the brains are different. All right, let's start at the front and work our way backwards. Okay. That one you can kind of see a little bit better. Okay, inverted U, Fornix, anterior lateral ventricles, septum pellucidum separating them. Caudate, it's kind of that bubble, half moon shape there. White matter, internal capsule. Over here in this gray matter would be the lentiform nucleus.
How would you mark the septum pellucidum specifically in that?
If I could stick a pin through that, then it'd probably be pretty difficult to do.
Yeah.
From a midline, it's
Yeah.
hard. It's sort of hard to do. All right. Yes, Patrice.
Where is the interior horn of the, is it?
Right here. So lateral ventricle, if we're in the anterior part of the brain, it's called anterior horn. If we're in the posterior, it's the posterior.
So, but we could just like the lateral ventricle and posterior.
I'd like you on this view to say anterior or posterior. That way I know you're orientated correctly, right? So if I were to mark caudate, I'd put a pin right like that. Okay? It's clearly in that top part of the gray matter. To mark internal capsule, it would be stuck in the white matter, which I know is kind of hard for you guys to maybe see, a little bit of a shade, that contrast. And then if it's stuck down here, right, then you know that that's the lentiform nucleus in gray matter. Okay? Okay. Same thing here. Here you can kind of see, again, lentiform, white matter, caudate, white matter, lentiform nucleus over here. Fornix. Okay. These ones that are kind of in the middle of the brain, I'm not going to use. I'm going to wait until I can see the fornix high and flat. When the fornix is high and flat next to the corpus callosum, these are the posterior horns of the lateral ventricles. In between the fornix and the corpus callosum. And then this is the T shape of the third ventricle, which makes this what? Thalamus and thalamus. Okay? Again, here you can just start, so this is hippocampus down here. You just start to see it coming around to make the hippocampus. We still have third ventricle here. If we flip around to the back side of this, now you really see how that hippocampal gyre is coming around there. We now have our pineal gland, and we have our hole for the cerebral aqueduct.
How do we differentiate between labeling hippocampus or hippocampal gyrus, or is that interchangeable?
It's the same thing.
Okay.
We call it hippocampal gyrus when it's on the outside of the brain, on the inside of the brain it's called. Yes, but
Will you ever be able to see the third ventricle and the cerebral aqueduct on the same cut?
no.
Okay.
Because the third ventricle narrows into that pipe. So once it becomes a pipe, it's a cerebral aqueduct, but it's still in a T shape. I mean, if I have it, I won't be tricky. Okay? You'll either see a T or you'll see a hole, right? Those are the identification marks. Yes, Becca.
So the caudate always goes to the anterior view and the thalamus becomes
Yes, absolutely. Caudate, lentiform nucleus, and internal capsule always goes to the anterior view. Thalamus always goes with third ventricle, posterior view. Okay? All right. Last thing that I want to show you is the human stuff. So, this is a nice slide of an unmarked coronal cut through the human brain. Okay? Longitudinal fissure that is separating the two hemispheres. Right? Here we have the corpus callosum in red, going across there. These are lateral ventricles. If there's a ventricle on the midline, it's third ventricle. So in the human brain, sometimes we don't get that really nice T shape because it's a little bit smaller than in the sheep brain. But it's the only ventricle that's going to be on the midline in the human brain that doesn't look like a little pipe. And I don't even think we have it. I don't think we have a good coronal cut of the cerebral aqueduct in the human slice. And this is sort of the section that I like to use to mark things. Okay? So here, this sulcus or fissure that's separating the top half and the bottom half of the brain is separating the temporal lobe from the parietal lobe. So that is the lateral sulcus or lateral fissure. And inside, this cortex that's inside the lateral fissure, that if you were looking from the outside, all you would see is that sulcus running along there separating temporal from parietal. Inside is the insular cortex, or insular lobe, that we couldn't do the other day. So what's in green here on either side is insular cortex. Sheep don't have that because they don't really have a temporal lobe. Okay?
Is that OR or AR?
AR. Insular. AR. Okay. Now, if we were to take a slice horizontally through the brain and look at it, the first thing we want to do is orientate anterior and posterior. How do we know where posterior is? Cerebellum, right? We also can see the ventricles. So here we have lateral ventricles, and what's in between the lateral ventricles? Septum pellucidum. What's right bordering the lateral ventricles, the anterior lateral ventricles? The caudate. So here we have caudate. This white fiber mass that's circled in green here is the internal capsule. Easier to see on this one, it's not marked. So we have caudate, white matter here that kind of makes like a V shape is the internal capsule. And then this lens shape out here, this dark lens triangular shape, is the lentiform nucleus. So when we're talking about an anterior cut in the sheep brain, we're slicing through like this, and we're seeing caudate, internal capsule, lentiform nucleus like that when we're slicing up and down, not horizontal. In the posterior part of the brain, for the human especially, this is not in the sheep, but in the human, the lentiform and internal capsule actually come all the way back to where the thalamus is. Now, how do we know this thalamus and not caudate? Well, right here in the middle is the third ventricle that's sort of dark in color, but that's a space, that's a void there that's dark. And that is a third ventricle, and on either side is the thalamus. And then you'll notice back here, this looks kind of like a funny part of the cortex. That's the hippocampus back here that's going into the temporal lobe, deep in the temporal lobe.